My talk is on networks in biomedical applications, where groupoids, and functional near-infrared spectroscopy. A network is a finite set of nodes, some of which are connected by the edges. It is a widely used tool in scientific research, including biomedical applications. Specifically, in neuroscience, networks are important for studying brain network architecture. Hybrid models are often used, physics-informed and data-based. Due to the complexity of the human brain, the researchers often use the functional connectome, which shows the connections between regions in the brain rather than individual neurons. The data comes from diagnostic equipment that acquires readings of neural activity via electrodes or other types of sensors, such as optodes for fNIRS. In mathematics, the accurate definition of a network is as follows. It is a finite set of vertices, say V, and a set of edges, E. Instead of using the term network, we call this pair of sets a graph. If each edge is directed, such a graph is called a directed graph. Otherwise, the graph is undirected. In these pictures, we have an example of an undirected graph and a directed graph, respectively. Rigorously, an edge is a pair of vertices. To visualize a graph, it is customary to think that a vertex is a point and an edge between two distinct vertices is a link, a line segment. It is permitted to have an edge whose source and target are the same vertex. Such an edge is called a loop. It is permitted to have two directed edges between two vertices represented by the arrows in the opposite directions. We need one piece of terminology. An undirected graph is called complete if there is an edge between every two distinct vertices. For example, the graph in the picture on the left is not complete because there are some edges missing. The graph in the picture on the right is complete. There is an edge connecting every pair of distinct vertices. The definition is similar for directed graphs, but we require that there is a directed edge in both directions. A weighted graph is a graph together with weights assigned to the edges. Generally, a weight of an edge is a real number. The choice of weights is determined by the specific problem or by the data set. If it happens that these numbers satisfy additional conditions and yield a metric on the graph, in other words, a distance function on the set of vertices, this might be very nice and provide additional mathematical techniques. A graph can always be embedded in a manifold, in the sense that its set of vertices is mapped into a configuration of n distinct points in this manifold. A smooth manifold can always be equipped with a Riemannian metric, which provides a way to measure distance between any 2 points in this manifold. Suppose the graph is equipped with its own metric. Can we embed this graph into a manifold symmetrically in such a way that the restriction of the manifold metric is the same as the graph original metric? This is a subtle question. Under some assumptions, the answer is yes. This is discussed in work by Graham and Winkler and also by Wren in these references. In this picture, we see a triangle graph, a graph with 3 vertices and 3 edges embedded into the sphere. This mathematical setup translates to the configurations of the sensors and channels in medical diagnostic procedures such as EEG or fNIRS. Electrodes or optodes represent the vertices. The channels, the transmitter-receiver pairs, correspond to the edges. The weights are the data from the channels. In this picture, the green dots are supposed to indicate the placement of the electrodes or optodes on the head. The discussion about embedding a graph into a manifold is relevant because there is some freedom or flexibility with regards to where exactly to place the electrodes on the skin. Even when there is a cap or a headset, the exact placement of each individual sensor can be somewhat adjusted. In the recent years, there is a huge amount of activity and publications devoted to the applications of network theory or graph theory in neuroscience, some of that reviewed in this paper from 2017, and of course, there is a well-known book by Sporns. fNIRS is functional near-infrared spectroscopy. It is a brain imaging technique that yields data near the cortical surface. It uses source detector pairs. The idea is based on tracking the changes in the hemoglobin concentration. It is a non-invasive, portable, and inexpensive technique. The recent 2025 paper referenced on this slide makes two fNIRS datasets publicly available. The first dataset was obtained from 17 healthy adult participants with a probe that contained 12 sources and 12 detectors, resulting in 24 source detector long separation channels and 8 short channels. The experiment was set up with 3 conditions: speech, noise, silence, or control. Each condition was presented 20 times. The data file from each of the 17 participants would consist of 60 rows like this. The discussion in that paper was centered around the following issue. Suppose different researchers analyze the same fNIRS datasets with their choice of methods, relying on their own perspectives. How does that affect results and hypothesis testing outcomes? A hypothesis typically would be of the form: speech stimuli evoke responses in a specific brain region, or speech stimuli evoke larger responses in one brain region than in the other brain region, and so on. Different approaches to analysis refer to data preprocessing, software choice, statistical methods, choice of parameters, or settings. To recap, there is an underlying connection between graph theory and the applied side of neuroscience. I would like now to make some observations that come from the mathematical thinking and may be of some value for applications. In math, given a set M, there is a notion of pair groupoid of M, which is the set of all ordered pairs of elements of the set equipped with additional algebraic structure. I will not write a full definition here, but let me say that the easiest way to understand this structure is as follows. Visualize such an ordered pair xy as an arrow from x to y. Two such arrows can be composed in the obvious way. If A, B, C are points in the set, and there are arrows from A to B and from B to C, then there must be an arrow from A to C, and we can multiply arrows so that the arrow from A to B times the arrow from B to C equals the arrow from A to C. There are other parts of the structure as well. There is a unit element, there is a concept of the inverse, there are source and target maps that extract from an arrow its source vertex and its target vertex. This works perfectly nicely for manifolds. If M is a manifold, then its pair groupoid is a manifold too, and we can think its points represent arrows between pairs of points on M. In case if M was equipped with a metric, we automatically get a metric on the pair groupoid. Since theoretically the pair groupoid of M is M cross M, the direct product of two copies of M, it's worth discussing how this compares with other familiar constructions. There is the set of configurations of 2 points on M. That is the set of all ordered pairs of distinct points of M. which is a proper subset of M cross M and not the full direct product. If M is the set of vertices of a graph, a finite set, we can take the product of this graph with itself. There are several definitions of graph product. In each case, the set of vertices will be M cross M. However, the edges of the product will depend on the choice of the definition. In this picture, I show three different definitions of a graph product applied to the product of the graph on two vertices and the triangle graph. We get three possible outcomes. Each is a graph with 6 vertices. The first one is the direct product of graphs. The one at the bottom is usually called the strong product of graphs. The strong product of graphs is defined as follows. Suppose X is a vertex in the first graph and X prime is a vertex in the second graph. Then the pair X prime is a vertex in the product of graphs. Suppose now Y and Y prime are vertices too in the first and the second graph respectively. Then in the product of the graphs, the condition for the vertices x prime and y prime to be connected by the edge is as follows. There is an edge between x and y, and there is an edge between x prime and y prime, or x equals y, and there is an edge between x prime and y prime, or there is an edge between x and y, and x prime equals y prime. I can prove that if a graph is complete, then the strong product of this graph with itself is isomorphic to the graph obtained from the pair groupoid. This statement requires a bit of an explanation, in particular what isomorphism means in this context. Intuitively, this procedure means that we switch from the graph to its set of arrows, which makes perfect sense for diagnostic procedures that involve channels. There may be no obvious relationship between functions built from data on the original graph and on its pair groupoid. For example, consider the triangle graph with the weights on the three edges that are positive real numbers that sum up to 1. The pair groupoid of the triangle graph is a complete graph on 9 vertices. The information theoretic entropy of the pair groupoid is not expressible in terms of the information theoretic entropy of the original triangle graph. I suppose I could run a numerical experiment to check the correlation between these values on the file from dataset 1 from the paper mentioned earlier. The pair groupoid of a set M is the space whose points are arrows connecting points of M. When M is a graph embedded into a manifold X, the pair groupoid of M is a subgroupoid of the pair groupoid of X. In diagnostic procedures involving transmitter-receiver pairs on humans, the pair groupoid of M contains the space of channels rather than points on the human body. It makes sense to consider the data calculated on the space of channels and also to consider M being embedded in a manifold due to variability of location of placement of electrodes or optoids. Work by Carl Pribham, Carl Lashley experiments, and other work in neuroscience relates processes in networks of neurons associated with learning to cumulative electric wave patterns in neural networks. And that intuitively may support the argument that structures such as groupoid may be useful and may be the correct way to study or to model how these networks function. It is possible to move further from the concept of a pair groupoid of M to the concept of the K groupoid of M. As I said, it is a direct product of K copies of M. These higher level structures have an intrinsic relationship with the multi-layer or multiplex networks. Multiplex networks are used to integrate data from different sources, for example, from EEG and fNIRS, as shown in these two recent papers. When working with data, spending time to extract anything related to the pair groupoid of a graph or product of this graph with itself means more computations. It adds complexity. However, it may possibly catch phenomena linked to the network level events or make those more clear and visible. The graph associated with a pair groupoid is complete, and it may not be sensitive to the structure of the original network in case if that was not complete. But we can make a complete graph out of the original graph by adding the missing edges, assigning to each a large weight larger than the maximum weight in the original graph, and study the insights obtained for the original graph from the pair groupoid of this modified graph. If you have any questions, then please feel free to contact me by email. Thank you for your attention.